This is one of these projects, just thinking back to like we started this work, I think in 2017, when, I remember we thought about how we're going to attack this molecule, and I'll show it in a minute, but it's one of those projects where if things pan out, it's going to be transformational. The aspirational route we're going for is really going to change things. So I'm honored to be here on behalf of the team to sort of share this really exciting innovation and story. So when we look at processes, our first goal is we want to develop a robust process, but we want to do it in the most sustainable manner as possible. We not only want to bring our drugs to patients, but we want to do it in a way where we sustain a healthy planet as we do that. And for those who know, doing organic chemistry can generate a lot of hazardous waste and can use a lot of energy. So it's an opportunity for us to really have a positive impact. One thing we do, and here's the sort of clinical route, this is what we call our original route. This gets the molecule into the clinic, gets it so we can figure out if this has the potential to be a drug. Initially, when we look at this, we like to step back and say, this is great, this is a nice way to make the molecule, but we really want to challenge ourselves. Is there a better way? Is there that sort of aspirational route? And usually what we find is that involves inventing new chemistry, new reactions, new ways to do this, right? And what I can show here, this was sort of state of the art, I would say, to make nucleosides when we started. And I tried to put a few metrics to point some of the liabilities. So at the start of this, it's 16 total steps. For those of you who don't appreciate that, each step, you run a reaction, you do some sort of isolation, you go to the next step. Each one of those steps takes a significant amount of time and energy to execute. So the goal is you want to reduce the steps as low as you possibly can to increase your efficiency and sort of your time manufacturing. And if you think about efficiency, because we're running 16 steps, we only get about a 14% isolated yield. So you can just imagine the amount of material we're wasting along in that synthetic process. And what that turns into is about 6,000 kilos of waste per 1 kilo of API we make. So API is your drug substance. That is the drug. So every time we want to make 1 kilo of a drug, we're generating 6,600 kilos of essentially hazardous waste, right? Not to mention the energy associated with generating that waste and then processing the API. Another limitation of this chemistry was the fact that we have to use protecting groups. What these mean, these are reactions that we have to install on our molecule to enable us to run the bond-forming transformation, and then we later have to cleave this protecting group off. So these are non-productive steps, right? These are not ideal. These are steps you're doing that are not building complexity in your API. And all 16 of these steps were run in a hazardous organic solvent. So our goal when we looked at this and thinking about sort of state of the art for nucleoside chemistry, one thing really jumped out at us, and that was this bond I have highlighted here in the glycosylation. And what was really concerning is after really years of trying to optimize this reaction, the best we could do is about 2 to 1. So if you imagine, that's throwing away around 33% of your material in that step. So you've gone 14 steps and you're going to throw away 33% of the material. So not ideal at that point. So what we stepped back and said, okay, we need that sort of step change. How else can we do this? We've sort of taxed these standard synthetic chemistry methods. And what we said is nucleosides, nature makes related looking compounds. Can we learn from Mother Nature? Can we see how it is done sort of biologically? And that led us to explore enzymes. And I'm pretty much going to go over multiple years of research in sort of a half a slide, so bear with me here. What we were able to quickly identify is an enzyme that did this glycosylation reaction, so that bond I have shown there, with essentially 100 zero selectivity, so perfect enzymatic selectivity. So that was the moment where we knew we were onto something and it was really exciting. What made this project even more exciting was that sort of aspiration with, wait, why stop at that bond? Let's use enzymes to make every single bond in this molecule, going back to simple commodity chemicals. What's even more exciting about that was we said, wow, can we do this in a single stream, essentially a single pot, one giant enzymatic cascade? So what I have shown here is you start with our triol shown there on the left, and you essentially, in one reactor with water, you basically run this sort of one-step cascade with a variety of different enzymes, each doing their job independent of each other. So in total, we're able to use 9 enzymes to make islatravir in essentially one sort of enzymatic cascade. And when you think about the impacts of that versus our green chemistry metrics, you can see we now have a 90% isolated yield versus our 14%. We have over 95% reduction in waste. So you take that back to that 6,600 is now 360 kilos of waste. As you might imagine, significantly shorter manufacturing process since we're doing one sort of enzymatic cascade versus 16 steps. We have no protecting groups. All 9 enzymes do GMP bond forming steps, so they make bonds that are in the molecule. And again, this is a single aqueous stream without any intermediate isolations and essentially no organic solvent. So a really exciting accomplishment for the team. And what I think was one of those moments where you get even more excited was when you think of where we started, you ask yourself, wait, why can't we do this more? Why can't this be a new blueprint for the way we make pharmaceutical agents? And I like to think of this, no pun intended, the way we evolve these enzymes is through directed evolution. And I always think of this as like the next evolution of pharmaceutical manufacturing. So a few metrics here just to sort of show them on a graphic form to point it out more. Essentially, as I talked about, 95% reduction in PMI, that's your overall process waste you can think of. Our global warming potential is down around 93%, and we've essentially, even though the process is run in water, the amount of water we've saved in this process from all the aqueous workups and stuff is over 97%, so really impressive. Now getting back to that sort of the blueprint for the future, these are some other drugs in our Merck pipeline, and as you can see, what we've learned from islatravir has totally changed the way we think about manufacturing pharmaceutical agents. And I would make the case in a significantly more sustainable manner, not only from the selectivity, the step count reduction, but also enzymes are biorenewable. They typically run in aqueous solvents. They inherently are catalytic and more sustainable. So really exciting. You can see the work here is islatravir. But if anyone had noticed in the literature, we built off of this to use an enzyme cascade to make molnupiravir, our COVID compound, a slightly different cyclic dinucleotide, our STING compound. We also did a 4-enzyme cascade. And for those of you who have been paying attention, we had some really exciting news recently about our PCSK9 inhibitor, shown there on the far right, enlicitide. This is a macrocyclic peptide, which I don't want to spoil the excitement because hopefully I'll be back here in the future for one of these awards, but we were able to, attack this molecule using enzymes and, to be quite frank, I don't think it would have been a viable product without enzymatic cascades. So very exciting to see how the work we've done in islatravir has really changed the way Merck thinks about manufacturing, and I truly believe it'll change the way other companies, continue to think about manufacturing drugs. And with that, this is definitely not doing it justice. This was a great collaboration across Merck and also with our colleagues at Codexis who helped on the protein engineering side. Just want to acknowledge all my fellow colleagues. If you think about doing something like this, there's the science to demo, you can do this in the lab, but then there's also those challenges of how do you take it from the lab to manufacturing, how do you do all the analytical release. So truly a great example of a collaboration across an entire company. And as you can see, we have multiple of the colleagues here, and that's just a fraction of them. So thank you to all of them. Thank you again for the award, and thank you for the opportunity to share this exciting work.